chemiosmosis hypothesis. This hypothesis was proposed by Peter Mitchell in 1961. This hypothesis explains the formation of ATP molecules in photosynthesis and respiration as well. According to this hypothesis, ATP synthesis during the photosynthesis is linked to development of the proton gradient across the thylakoid membranes. Development of the proton gradients occurs according to following mechanisms. How proton gradient develops? When PQ transports an electron, it removes the H plus from the stroma and transfer to the lumen. And NADP takes up the proton and become NADPH. Whereas in thylakoid lumen, when PQ transfers an electron, it removes the H plus from the stroma to lumen, and splitting of water molecules occurs inside the membrane, so protons start accumulated. Water molecule splitting and gives the H plus ion electrons and oxygen. Splitting of the water molecule takes place on the inner side of the thylakoid membranes. Thus, protons are during the photolysis are accumulated within the thylakoid lumen. With the transport of the electrons through the photosystem, thus protons are also floated across the thylakoid membranes. It occurs because in PSI1, electrons transported through the NADP, a proton carrier. Hence, NADP removes a proton during the electron transport. Photolysis of water also requires minerals such as manganese, chlorine, calcium. Hence, NADP removes the protons from the stroma during electron transport. As protons are released into the lumen of the membrane. Thus, the number of protons are decreased in the stroma, increased in the lumen. Creates the proton gradient across the thylakoid membranes. This proton gradient is broken by the transmembrane channel known as CFO CF1 ATPase. CFO means C means a coupling factor. This transmembrane channel consists of an ATPase unit. It catalyzes the formation of the ATP from ADP and inorganic phosphate. When H plus ions moves from the inside to outside, a configuration change occurs in ATPase unit. Due to this, it catalyzes the formation of the ATP from ADP and inorganic phosphate. 3 protons are required for the formation of the 1 ATP molecule through the F0 F1 particle ATPase unit. This mechanism of the ATP formation is known as chemiosmotic ATP formation. This takes place in photosynthesis and respiration as well. Complex fifth or F0 F1 complex. This complex has components, namely first one F1 ATPase, second one F0 component. When we draw on the board, we can see that F1 ATPase peripheral membrane protein contains 8 subunits. These are paired 3 alpha, 3 beta, 1 gamma and 1 epsilon. F4 component integral membrane of protein attached by means of its gamma subunit. One more subunit will be, that is a delta subunit. There is no activity. F1 component consists delta 3 component of 3 copies of alpha subunits, 3 copies of beta subunits resting on gamma and epsilon and its subunits. F0 particle contains the 1 copy of A, 2 copies of B, 10 copies of C subunits. F0 components all are embedded in the inner membrane, which is lipid layer is hydrophobic, whereas F1 which are protruding from the inner membrane is hydrophilic, which is embedded in matrix. In F1 ATPase, delta subunit has no catalytic activity. Actual ATP synthesis takes place in the beta and gamma axial. ATP binds between the alpha and beta. Gamma is in the form of bent axial. It is not a straight orientation which are alternating each other. It is surrounded by 3 alpha beta subunits which are connected by the nucleotide binding catalytic sites. F1 part often compared to the lollipop. F0 part located within the inner membrane of the thylakoid serves as a rotor-like motor and channel for transport of ion from the intermediate space to matrix. It comprises the 10 C subunits make up rotor-like motor. 1A subunit which serves as a H plus ion channel. 1B subunit which extend the inner membrane into the matrix to stabilize the catalytic subunit of the F1 port which contact the delta subunit. F1 located in the matrix and serves as a axial and catalytic port of the ATP synthase. The axial comprises the 1 gamma subunit, 1 epsilon subunit which are connected to the C subunits which are rotor-like motor and extend up to the catalytic port which comprises the alpha beta subunits, which starts 1 H plus ion binds to C subunit which causes the rotor-like motor to rotate the 120 degrees. That H plus ion then exits intermediate space through the A subunit which acts as a ion channel. The rotation of the rotator-like motor causes the axial to rotate the 120 also, which causes the one another subunit loosely binds ATP and free phosphate. Once the hydrogen ion makes the complete rotation, it makes through a channel in the stator of the ATP synthase. The catalytic knob consists of several subunits, including the 3 catalytic subunits that can be found in 3 different states: ATP and phosphate binding state, and ATP forming state, and ATP releasing state. The next ion then causes the rotor-like motor and axial through the rotate another 120 which promotes the tight binding of ATP and free phosphate. third and last H1 ion then causes the rotor like motor and axle to rotate another 120 degrees, which promotes the formation of the ATP and release away the ATP. In ATP synthase, beta and gamma rotates, after which a new ADP and free phosphate loosely bind to the unoccupied subunit. This process continues to produce the ATP for every 3 H plus from intermediate space to into the matrix. Then in photosynthesis, whereas lumen to stroma across the thylakoid membranes.
Binding change mechanism for ATP synthase with ATP synthase. It is otherwise called as flip-flop mechanism. This mechanism was developed by the Pulse Boyer. It works like a turbine. Let's see, as of now, how ATP synthase carries out this mechanism. Although the alpha subunit contain in a bound ATP, they do not release it and they do not actually participate in this reaction. The beta subunit, however, can bind to the ATP and inorganic phosphate, synthesize the ATP and release into matrix at any time. So, beta subunit can exist in one of three distinct states. There are three states. One is loose state, open and tight state. The rotation of the Y subunit allows the interconversion of the beta subunit from one state to another. Notice that all beta subunits exist in distinct states. It implies that any two subunits will never exist in same identical states. A rotation of the Y subunit of 120 degrees in the counterclockwise direction converts the beta subunit in tight state into the open state. This releases the beta subunit and allows it to release the ATP. Once the ATP is released, a new set of ADP and inorganic phosphate will enter the open beta subunit. Another rotation of 120 degrees in the counterclockwise direction will lock the reacts in the by converting the open state into the loose state. This description is known as binding change mechanism.